Study the following statements. Indirect ELISA is used to detect antibodies in the sample. Second one, in sandwich ELISA, the well is coated with primary antibody. The T cell play an important role in the allograft rejections. Xenotransplantation is often an extremely dangerous type of transplantation. Now, out of these, what are the correct statements? Now, the first statement, as we observe the indirect ELISA, suppose a person has been suffering from HIV virus. That means a person has been suffering from AIDS. He is doubtful regarding the AIDS. Now such person is going to approach a doctor. Now the doctor going to observe or conduct what type of test? Indirect ELISA. Because of the indirect ELISA, we are going to observe the antibodies. It is not a much costlier process, because a person, as we observe one person, into his body HIV virus entered. Against HIV virus in his body, slowly antibodies are going to develop. How much time is taken for the development of antibodies against HIV virus? 3 or 4 months. After 4 months, if the AIDS patient is going to approach a doctor, he is going to conduct indirect ELISA. Because of the indirect ELISA, we are going to observe the antibodies against HIV virus. It is not a costlier treatment. What about the sandwich ELISA? It is direct ELISA. By this test, we have to identify the antigen. Now, as we observe the direct ELISA, now this one we call it as a vessel. Now this is the bottom. Now we add the first dose of primary antibody. First dose of primary antibody. Okay. Now the next one. This is first dose of primary antibody. First dose of primary antibody. Now we add the blood sample. Blood sample of AIDS patient. Suppose the person has been suffering from AIDS. We imagine the person has been suffering from the AIDS. Now his blood sample is added. If he has been suffering from the AIDS, in his blood plasma antigen present. Now this is the antigen. This dot indicates the antigen, AN. So the antigen attached to first dose of primary antibody. Now we add second dose of primary antibody. This is second dose of primary antibody. Now this is one antibody, this is another antibody. This appears to be a bread piece. This appears to be another bread piece. Between two bread pieces, what is present? Curry is present. In the same way, in between these two primary antibodies, what is present? The antigen present. Now it is called the sandwich ELISA. Now we add. This is the first dose of primary antibody. This is the antigen. Second dose of primary antibody. Now we add the enzyme linked secondary antibody. This is enzyme linked secondary antibody. Now in this test, how many antibodies used? 1, 2, 3. Now we add reagent. That reagent is chromogen. If chromogen is added in this vessel, if antigen present in the blood sample of AIDS patient, automatically the color changes. If color changes automatically in the blood sample of AIDS patient, what is present? The HIV virus is present. Now the test is said to be positive. In the blood sample of AIDS patient, if there is no antigen, if we add the chromogen, there is no color change, then it is said to be the negative. So the second statement, the sandwich ELISA, the well is coated with, now you observe the diagram, the well is coated with the first dose of the primary antibody. Now the third one, the T cells play an important role in the allograft reactions. Suppose from one person or in our body, suppose kidneys are damaged. Now the kidneys are replaced by another person's kidney. In the initial stages, our body does not allow the transplanted organ. In order to prevent the rejection of transplanted organs, we go for the monoclonal antibodies treatment and T cell treatment. They play an important role. They prevent the rejection of transplanted organs. So the third one, the T cell play a crucial role in the allograft rejections. And the fourth one, genotransplantation. That means transplantation between two different species. Suppose in our body, regarding the heart, in the heart valves are present. One is tricuspid valve, bicuspid valve and semilunar valves are present. Suppose any valve is damaged, such valve is replaced with the valve present in the pig body. That is a different species. That means from the pig body we are taking the valve and that is arranged in our body. We are Homo sapiens. Our species is different. The pig body species is different. The transplantation between two different species, that is much dangerous. It is very rare. So whenever the transplantation taken place between the two different species, our body generally may or may not be the accept. So because here the transplantation between the two different species, so that the genotransplantation is often an extremely dangerous type of the transplantation experiment. So the four statements are correct statement. Now the next question, which one of the following is used to accurately measure the density of the bone in evaluating the osteoporosis? Now as we observe in our body, this is one bone. The formation of holes in the bone, we call it as osteoporosis. Now we want to know the density of the bone, whether it is the thick or thin. To know the density of the bone, what type of test is conducted by the radiologist? That we call it as, one option we call it as CAT. The second one is X-ray. Third one EEG. And the fourth one is ECG. The ECG associated with the working of heart. What about the EEG associated with the working of brain? And what about the X-ray? A general, that means a general observation in emergency conditions. And the CAT, computerized axial tomography. With the help of the CAT, we can assess the density of the bone, whether it is the thick or the thin. The density of the bone can be identified with what type of the radiography, that we call it the CAT, computerized axial tomography. The answer is the first one. The next, in ECG, electrocardiogram, enlarged Q wave indicates. Now as we observe the ECG, electrocardiogram, that is instrument, electrocardiograph, that is the graph paper coming from the instrument. Now as we observe the electrocardiograph, in the graph paper it is a baseline. This one we call it as the baseline. Based on this baseline, the graph is drawn by the instrument known as ECG, electrocardiogram. Now this one we call it as the P wave. This one we call it as the P wave. And this one we call it as the Q R. And this one we call it as the S. And this is called the ST. And this one we call it as the T. P wave, QRX complex, ST segment, and T wave and continuity of T. Now this entire one we call it as the baseline. Now as we observe this diagram, this is the graph. As we observe this graph, this is the P wave. Above baseline P wave present upward deflection. Q below baseline downward deflection. And R above baseline upward deflection. S below baseline downward deflection. And it is on baseline ST. And the T above baseline upward deflection. Now this one we call it as the P wave. And this one we call it as the P or interval. I means interval. Now this one, the QRS. This indicates the QRS complex beginning, ending of QRS. And this is ST segment. This indicates the end of QRS complex and beginning of T. Now this is the center of T. Now you observe here a small elevation. The elevated part of T, that we call it as the apex. Now from the beginning of QRS and in the center of T, this we call it as absolute refractive period. Now you observe the diagram. From the beginning of QRS and from the center of the T, that is called absolute refractive period. Now this half we call it as the relative refractive period. Now again this one, you observe the graph from this to this. Now here absolute refractive period plus the relative refractive period is equal to the QT. What is meant by QT? Absolute refractive period plus relative refractive period. And now this one, the time of QT, you observe the graph. The time of QT is 0.4 seconds. And this one, the time of this part is called 0.2. And the time of this remaining part, 0.2. So what we learned in the working of heart? One heartbeat completes 0.8 seconds. One beat completes 0.8 seconds. Now the 0.8 seconds are classified into auricular systole and ventricular systole, complete cardiac diastole. Auricular systole lasts for 0.1, ventricular systole lasts for 0.3, complete cardiac diastole lasts for 0.4. So you count 0.8 seconds. One beat lasts for 0.8 seconds. 1 minute, how many beats? Minimum 70, maximum 72. Now as we observe the heart, the 4 chambers. These are 2 auricles and ventricles. Now the P wave. The P wave indicates the auricles depolarization. Whenever the auricles are filled with blood, the 2 auricles will undergo contraction. That is called auricular depolarization. That is called the P wave. What about the QRS complex? The ventricles depolarization. Whenever the auricles contract, the blood enters into ventricle. Now the ventricles contract. The ventricular depolarization, QRS complex. What about the T wave? That indicates the ventricles repolarization. That means the ventricles come to original state. Now, based on this graph, we have to discuss some questions. Now, in the ECG, enlarged Q wave. Now, this is the QRS. In a healthy person, if we take the graph with the help of ECG, we are getting like this. In this graph, if any abnormalities are going to observe, we have to discuss some diseases. Now here what we are asking, in ECG, enlarged Q wave. Now this is the originality of the Q wave. If the Q wave enlarges, that indicates the myocardial infraction. What is meant by myocardial infraction? That is nothing but the heartbeat. Now here the answer is, so the enlarged Q wave, this is a general ECG of a healthy person. Suppose if we are going to consult our physician, he has to take the ECG with the help of the electrocardiogram. Now this is the general graph of a healthy person. This is the P wave and this is the QRS complex and this is the T wave. If this Q wave enlarges, that we call it as myocardial infraction. Myocardial infraction is nothing but the indications of the heartbeat. That means heart stroke. Now the next one. Hypoxia due to myocardial ischemia is indicated by. Now this is ST segment. Now in this diagram, that means this is a healthy person ECG. In a healthy person ECG, as we observe the ST segment, this line is on baseline. Now here we are asking, hypoxia due to myocardial ischemia is indicated by, as ST segment Now from the baseline, the ST segment depressed. If ST segment depressed, that we call it as myocardial ischemia. What is the symptom of the myocardial ischemia? The heart wall does not get the sufficient oxygen. So here hypoxia, less oxygen is supplied to the muscles of heart. Hypoxia due to myocardial ischemia is indicated by, the answer is depressed ST segment. Now the next one. Brain aneurysms can be detected by observing. Now what is meant by aneurysm? This part we call it as the skull. In the skull, one part we call it as the cranium. In the cranium, what is present? The brain is present. Now the blood vessels supply blood to the brain. What is meant by aneurysm? This one we call it as the blood vessel. As we observe the volume of blood vessel, if the volume of blood vessel increases, that means the swelling of blood vessel, the enlargement of volume of blood vessel, that we call it as aneurysm. Associated with the brain aneurysms can be detected by what type of scanning? That we call it as the magnetic resonance image, that we call it as MRI scan. With the help of MRI scan, we can detect the aneurysm of the blood vessels. The blood vessels supply blood to brain. If the volume of the blood vessel increases or the blood vessels enlarge, that is detected by what type of scan? MRI. Now the next one. In the ECG, relative refractive period is, now this one we call it as the T wave. Now this dot line indicates the center of T line, that is written as apex. Now you have to observe this one, that is QRS beginning and ending of QRS. You have to observe the diagram. This is the QRS. The QRS complex indicates the ventricles depolarization. Now this is absolute refractive period. What is meant by absolute refractive period? From the beginning of QRS and in the center of T. The center of T we call it as apex. Now you observe, what is meant by absolute refractive period? The beginning of QRS complex and the apex of T. Now this is the remaining part of the T. This is called the relative refractive period. Now here, in the electrocardiograph, relative refractive period is the last half of the T wave, this one. This is called a relative refractive period. Now, relative refractive period, absolute refractive period is combinedly termed as, that we call it as QT. Now here the answer is the next one. Transplantation of complete set of organ is known as, suppose in our body the kidneys are completely damaged. The kidneys are completely replaced by another new organ. The transplantation of the complete set of organ is known as domino. The answer is, now already we discussed, that is isograft transplantation between genetically identical, generally with reference to twins. What about the autograft? One person in a person body from one place to another place transplantation. Allograft, same species genetically not identical. and split transplantation. One organ is divided and is given to two persons, split. But here, domino, the organ is completely replaced by another new one. The next one, blocks in the blood vessels, aneurysms, arteriosclerosis, epilepsy diagnosis, enzyme coupled antigens. And now blocks in the blood vessels. Now in our body, as we observe the blood, this part we call it as the blood vessel. In the blood vessel, the blood is passing. In the blood vessel, here and there, the blood is going to clot. We want to know the blood clotting inside the blood vessels. Now the first one is blocks in the blood vessels. How it can be known? Digital subtraction angiography. What is meant by angiology? The study of the blood vessels. Inside the blood vessels, the blood is passing. In the blood vessels, to know the clotting of the blood, we go for the digital subtracting angiography. Whenever it is connected to intensifier and cathode ray tubes, automatically we are going to know in the blood vessel where the blood is clot. Aneurysms, already we discussed. Aneurysms, swelling of the blood vessels. How it can be noticed, how it can be detected, with the help of what type of scan? The MRI. And arteriosclerosis, how this one is going to occur? Prolonged PR interval. Now this is the PR interval. This is PR interval of a healthy person ECG. If this PR interval, that means the beginning of P wave and the beginning of the QRS. If this gap is going to increases, that is enlarged PR interval, what is going to happen? Arteriosclerosis. In our body, arteries, that means in Telugu called dhamanulu. As we observe the arteries, the walls of arteries are thickened. Arteriosclerosis. In the walls, the fatty material, the cholesterol material, all are accumulated. Slowly the walls of arteries increasing. So the lumen slowly becomes the narrow. In the arteries, this is the lumen or the cavity, and this is the wall of artery. Whenever the cholesterol and other materials are accumulated in the blood of walls of arteries, slowly the lumen is narrow. There is no proper supply of blood to the muscles of the heart. That we call it as arteriosclerosis. That is indicated by enlargement of PR interval. Now, epilepsy. What is meant by epilepsy? A falling sickness. A falling sickness associated with the working of the central nervous system. Epilepsy, falling sickness, but it is not treated as a fits. It is epilepsy, only falling sickness. That means if the central nervous system or the brain is not properly working, the patient has been suffering from the falling sickness, that is epilepsy, but not the fits. The one who has been suffering from the fits, the patient, if the suffering from the fits, in his body, as we observe, the hands and the legs are exhibiting this type of the movements, and the saliva comes out in the form of the foam. The one who has been suffering from the fits. But here the epilepsy, falling sickness, that means in his body the central nervous system or the parts of the central nervous system are not properly working. How it can be noticed? With the help of EEG, electroencephalograph, but not ECG. ECG associated with the working of heart. EEG associated with the working of the brain. Now, enzyme coupled antigen. To know the presence or absence of HIV virus in the blood sample of the AIDS patient, we go for the direct ELISA, that is enzyme coupled antigen. Now the next question. In X-ray imaging, soft tissues of lungs appear in shades of grey and air appears black. Now as we observe the X-ray film of one patient, there we have to observe the grey color and the black color. Now the reason, pulmonary organs absorb much of the X-ray during the X-ray examination. No. In our body, as we observe the bones etc., they are dense parts. They absorb more and more X-ray radiation. But what about the soft organs? One example is the lungs. As we observe the soft body parts, through the soft body parts the X-rays are passing. But as we observe the thick parts, through the thick parts the X-rays does not pass. They absorb the X radiation. But through soft parts the X-rays are passing. But here assertion, in X-ray imaging, soft tissues like the lungs appear in shades of grey because through the soft parts the X-ray radiation passing. So the soft parts appear to be grey and air appears as a black. Between the organs and the systems, if air is present, that appears with the black shades. So A is correct. Reason, pulmonary organs, they are the soft organs, absorb much of the X-rays. No, through the soft organs X-rays are passing, so they appear with the grey. Between the organs, if air is present, that appears with the black color. So here the reason is wrong, the assertion is correct. Now the next one. Read the statements regarding the X-ray radiography. There is always much more chance of cancer for radiation. Whenever our body is exposed to the X-ray radiation, there are chances of getting the cancer because the X-rays having the power of ionization. But we are not getting the much cancers. There are no much chances of getting the cancer. We are having the chances getting the cancer, but not much chances. The second one, the wavelength of X-rays is in the range of 10 to 0.01 nanometers. Exactly, it is correct. The third one, flow of blood in the blood vessels can be studied in angiography. Suppose in our body the blood vessels are present. If the blood vessels with the help of X-ray, we know or we observe the image of the blood vessels. But in the blood vessels, the flow of blood is not identified with the help of X-ray. And we are unable the clots which are present in the blood vessels. How? The X-ray is associated with intensifier and cathode ray tube. Then we are getting the sounds of the passing of the blood. And we have to observe the blood clotting in the blood vessels. All these are observed whenever the X-ray is associated with. That means the imaging of X-ray is associated with intensifier plus cathode ray tubes. We are getting the cancer, but no much chances. We are having the chances of getting the cancer when our body is exposed to the X-ray radiation. So that the answer is. Now the next one. coronary ischemia is indicated by, now as we observe in our body, here the heart is present. Name the artery that supplies blood to the heart muscles, the coronary artery. Whenever the coronary artery is damaged, that is replaced by another blood vessel. Whenever the coronary artery is blocked, there is no proper blood supply to the heart muscles. We are getting the symptoms regarding the heart. Now here the question is, coronary ischemia is indicated by, now this is ST segment. Because of the depression of ST segment, now in a healthy person ECG, the ST segment is on baseline. If it is depressed, that is one reason. Depression of ST segment, the QT, if the QT time is lengthened, these two indicates what type of disease? Coronary ischemia. There is no proper blood supply to the heart muscles. Now the next one. One of the following is not a benefit of X-rays. In the second year topics we discussed the benefits of X-ray radiation and what are the harmful effects of the X-rays. Both are discussed. Now here we are asking, one of the following is not a benefit of X-rays. Now, no radiation remains in the patient's body after X-ray examination. Suppose we are going to approach a radiologist by taking the X-ray film. Our body is exposed to the X-ray. Now we are getting the X-ray photograph regarding our body parts. After completing the taking the X-ray film, there is no question of X-ray radiation in our body. So no X-ray radiation remains in the patient's body after X-ray examination. No X radiations in our body after completing the examination. Second one, X-ray radiography is useful in emergency diagnosis and treatment. Suppose we are getting the lung problem, we are getting the heart problem, we are getting any problem regarding our body. To know the problem immediately, we are going for the X-ray. If any defects are going to observe in the X-ray film, then step by step we go for the CAT, we go for the MRI, and we go for the remaining scanning. But initially, to know the problems regarding our body, we go for the X-ray. Third one, X-ray usually has no side effects. If it is if our body is continuously exposed to the X-ray radiation, what is going to happen? The X-ray radiation having the ionizing properties, so we are getting the chances of cancer, but not much chances. Now the fourth one, X-ray radiation always has a slight chance of cancer. So the last one is not a benefit. The first three are discussed at the benefits of X-ray radiation. The last, if our body is exposed to the X-ray radiation, because of the ionizing properties of the X-rays, we are getting the chances of the cancer. That is not a benefit. That is the harmful effect of the X-ray. Now, the students, with this class, our EAMCET program is completed. Now again, we have to repeat briefly. Regarding the first year, we are getting the 20 bits, and second year we are getting the 20 bits. Now on May 10th, we are going to attempt the AP Medical EAMCET examination. This examination regarding the old syllabus, this is the last paper. From next year onwards, we are preparing NCERT books. The CBSE or NCERT syllabus is a beautiful syllabus. It is very, very simple. We can prepare up to the mark. And on 5th, we are going to attempt the NEET examination. Regarding the NEET examination, each paper having the 45 bits. And each corrected question having the 4 marks. If the question is wrong, 1 mark is going to lose. And no negative marks in the AP medical examination. But we are having the negative marks regarding the NEET examination. And our CAB TV provided an excellent coaching for the students who are going to attempt the NEET examination as well as the EAMCET medical examination. In all these days, we are discussed the botany bits and zoology bits, physics, chemistry, mathematics. And every day we are discussed less than 40. In some classes we are discussed more than 40. In some classes we are discussed 80 bits. And all these bits are very, very important and repeatedly asked in the previous EAMCET examinations. And in all these days we discussed each and every topic. Regarding the first year zoology, we discussed the basics, animal organization, gender characters and classification, man and biosphere, earthworm, cockroach, animal associations. In the second year, we discussed the gender characters and classification of chordata, pisces, amphibia, reptiles, aves, mammals. And we discussed the rabbit functional anatomy 1 to 3, evolution, genetics, aquaculture, poultry, and the biomedical technology. And what we discussed and what we learned in all these days, the chance given by our beloved the CAB TV. Now the CAB TV is going to conducting the classes for the incoming. That means for the first year students and second year students also. In the coming days, the CAB TV providing the classes, e-classes for the first year, second year, and the NEET examination and the EAMCET examination. Once we sincerely approach these classes through TV program, definitely we are going to get the success. And here one more thing is, once we are going to confidently attempt the examination, no doubt we are going to get the seat getting rank. Regarding the botany and zoology, in AP medical examination, botany 40 bits and zoology 40 bits. Out of 80, if we are going to get 75 plus in the biology and 30 or 35 marks in the chemistry, in physics we are going to get 15 to 20 marks. Definitely we are going to get the seat getting rank. For reservation candidates, for ST reservation candidates, if they are going to get out of 160 between 80 and 90, they are going to get the seat getting rank. For SC reservation candidates, out of 160, if they are going to get 95 marks, between 90 and 95, they are going to get the seat getting rank. And for the BCA and those reservation candidates, they are going to get 110 plus, 110, between 110 and 115, they are going to get the seat getting rank. And unreserved candidates, open category students, they are going to get 120 plus out of 160. So while attempting the EAMCET paper, you don't do any mistake regarding the well-known question. For example, regarding the zoology, we are going to attempt the 41 question. Now we are attempting the 41 question. Regarding the 41 question, we know the answer. And we go for the 42 question. If we are not getting the answer regarding the 42 question, don't waste the time for that question. Go for the next, go for the next, go for the next. So the time is saved. If we are doing the questions, if we are doing well-known questions first, the time is saved. And regarding the zoology paper, in the first year, the Periplanet Americana definitely covers 3 bits, and Feridima possuma covers 3, maned biosphere 4 bits, animal associations 3. What about the regarding the second year? We already discussed many times. The rabbit covers the 6 bits, genetics 3, evolution 3, and the remaining aquaculture, poultry, biotechnology parts, and the biomedical technology, and all these are the miscellaneous topics. From these miscellaneous topics, we are getting not less than 4 marks. Regarding the second year general characters and classification, we are getting second year general characters and classification 4 marks. First year general characters and classification, we are getting the 4 marks. Now in all these days, through TV program, we discussed important bits. Now again, the remaining last 1 week, you refer again the recorded programs. If you refer the recorded programs, automatically regarding all subjects, we are getting the questions again and again. So once we repeat more and more times in a short period, definitely we are going to get the success. So in each program we discussed not less than 40 bits. For example, 20 days or 25 days we discussed. 25 into 40, that means near about 1000 bits are discussed with reference to each subject. These are the more than enough. The CAP provided a better opportunity for us. Through this TV program, we learned A to Z in a short period. All topics are discussed in a bitwise manner, not in a theory. So once you revised all these bits regarding all these subjects, definitely we are going to get the success. More than 100% we are going to get the success. In the main EAMCET examination, the paper is fair. AP medical examination strictly with reference to the Telugu Academy textbook. If you thoroughly go through the Telugu Academy textbook and through this TV program, we discussed many questions, and all these questions refer 2 or 3 times or many a time, we are getting the success. Good luck students, meet again.